So hello everyone and welcome to our webinar, Golden Years of Semi-Automated Patch Clamp. So a warm welcome on behalf of the Nanion team. I am Marketing Associate Jaymar Portillo, and before we start, I would like to give you some general information about the webinar. By default, your video will be off and your audio will be muted. So this webinar would be recorded, as you probably have already seen. And by staying in this session, then you are consenting to being recorded. This webinar would be also later available on demand. For the Q&A sessions, they would be available after each speaker's presentation. And you can write your questions along the presentations. So we invite you to write them in the chat. But if you would like to formulate that yourself to the speaker, you can, after the presentation, raise the hand in the chat, and then the moderator will call your name and you can ask it yourself. And then at the end of the webinar, we will have a survey available for you, and we will appreciate very much your feedback. So a little information on the timeline of the webinar. We will have the first presentation by Dr. Patrick Mumm. Then second, we will then welcome Parsa Arman. And the third speaker of the webinar would be Elizabeth Kim. All right. So without further ado, we can then get started with the webinar. So it is my pleasure then to introduce you to my colleague, Dr. Patrick Mumm. Among his responsibilities here at Nanion, he is the product manager of the PortaPatch, of the product family of the PortaPatch. And he has been with Nanion for over 10 years. And it is our pleasure to welcome him today to tell us more about semi-automated patch clamp. So without further ado, the floor is yours, Patrick.
Thanks, Jaymar, for the kind introduction, and welcome everybody to this today's webinar. I just need to share my screen before I can get started. And yes, I just wanted to give you a kind introduction into the history of the PortaPatch and how it all started. So as you all know, the patch clamping is still the gold standard technique for studying the function of ion channels or electric currents transported by ion channels. With this technique, you place a tiny glass pipette on the cell membrane, you form a very tight seal and then gain electrical access by rupturing the membrane. The drawback of this technique is that it is a rather low throughput, it is very time consuming, and it requires significant training and technical capability, because you need to find your pipette tip under the microscope, you need to place it on the cell membrane, you need a vibration damp table, a rather big Faraday cage, and in general, quite a complex setup, which costs quite a bit of money. But in the 1990s, it became apparent that there was a need for a higher throughput in patch clamp technique, because several drugs were withdrawn from the market due to their side effects. And with this also mirrors a change in regulatory guidelines for drugs that came on the market. So there was a need to check those drugs on toxicogenic side effects or their potential on prolongation of QT intervals, for example, which resulted in the 2013 founding of the CIPA initiative, which will form a new paradigm of assessing pro-arrhythmic risks of new drugs. So in the late 90s, a bunch of companies were founded that were trying to put out the first automated patch clamp instrument on the market. So they all had similar approaches. Some were putting cells into a pipette, some were getting rid of the pipette, some were using loose seals instead of giga seals or population patch clamp approaches, but the most promising approach was getting rid of the pipette by using a planar glass substrate. The first thoughts about that were developed in the 90s, by 3 Ukrainian scientists. And to my shame, I didn't know that until about a couple of days ago, to be honest. But the first and the initial thought behind it by getting rid of the pipette is because no pipette, no hustle. You don't need the costly and heavy equipment like a vibration dam table, a huge Faraday cage. You do not need to pull your pipettes on a daily basis. You don't need a microforge and all that stuff. And the first successful recordings of cells on the planar glass chip were described by Niels Fertig, Robert Blick, and Jan Behrens in 2002. Niels Fertig, the good-looking man on the right side of the slide, and that led to the forming the foundation of the company where I work now almost a little bit over a decade. And when you do the math right, this was 21 years ago, and a year after the foundation of Nanyang, the world's first semi-automated patch clamp system saw the light of day, the Porter Patch, which is still until today the smallest patch clamp rig available on the market. It can record from one cell at a time, and it's very easy to use. It's very tiny, it has a very small footprint, but basically can do a lot of the things which you can do on a conventional rig, taking away the hustle of making pipettes and having that time-intensive training. So it's very easy to use. You can generate data basically on the first day you're working with it after having a short introduction. You can record voltage and current clamp on it like on a conventional rig. You can record whole cell and single channel ion channel activity. Due to the small volumes involved in the Porter Patch, there's a very versatile liquid handling. You have several add-ons, which I will mention in a little bit more detail later on. You can use it with the most amplifiers available on the market. You can use light as a stimulus for your cells, and we have consumables which are produced in-house, which can be customized. You also have the flexibility like you would have it when you make your own pipettes. Since the Porter Patch has been released, it has been adopted all over the world in over 20 countries, almost 100 universities and over 100 groups within academia and industry alike. And over 200 papers have so far been published with data of the Porter Patch, quite some of them in even high impact journals like Nature or Science. And the Porter Patch has shown his flexibility with a wide scope of different cells which were recorded on the Porter Patch, like primary cells or stem cell derived cells like cardiomyocytes or red blood cells. A lot of the cell lines that are available like HEK cells or CHO cells. You can record organelles on the Porter Patch like lysosomes or mitochondria, and even giant unilamellar vesicles. And with the variety of the cells you can record comes an even bigger variety of the targets you can record, like voltage-gated ion channels, like families of calcium and sodium channels, ligand-gated ion channels like ASICs or nicotinic acetylcholine receptors, and a lot more other channels and reconstituted proteins which you can put in your GUVs. The nice thing of the Porter Patch that you can tailor it to your needs by the nice add-ons that are available for the Porter Patch, like the external perfusion, where you can perfuse the external side of the cell you're recording with up to 8 different solutions to do precise dose response curve or activate ligand-gated ion channels. In this external perfusion, you can incorporate the temperature controls, so you can activate targets which are accessible for cold or heat activation, or if you want to record at physiological temperature. We have an internal perfusion where you can perfuse the cytosolic side of your cell with this tiny microcapillary which sits underneath the chip. We have a dedicated lid which fits on the Porter Patch, which can handle up to 3 different light sources when you want to stimulate channels like channel rhodopsines or work with caged compounds. And we even have a microscope slide which you can put under an upright microscope so you can see the cell you're recording in terms of checking your transfection rate or combining it with calcium imaging or things like this. And 20 years ago, the Porter Patch, as it was released, looked like this. And due to its proven success and robustness, we didn't change a lot in the last 20 years, so that the model now basically looks the same with a tiny facelift. But of course, the Porter patch is not there where the story of Nanion stops. It was the starting or the start of Nanion investigating and developing more instruments and broadening the portfolio, like fully automated multi-channel automated patch clamp devices like the patch liner or the synchro patch, or adapting new technologies like the surfer for transporter research, or the cardiac side to have a look at contractile cells like iPSC-derived cardiomyocytes. Or we have a dedicated instrument family dealing for bilayer recordings. And Munich nowadays has offices all around the globe. Like we have offices in America, in Germany, in Denmark, in China, and in Japan. And we want to be your one-stop shop for membrane science all over the globe. So if you're dealing with automated patch clamp cell analytics or membrane biophysics, I'm pretty sure we can be a good partner for your research. So that was it for a short intro from my side. And now I'm very curious to welcome our first speaker, Parsa Arman from the University of Texas. He has a background in biology and neuroscience and is currently a PhD student in the lab of Fernanda Leza. And we are very curious to hear something about IFG F14 peptide derivatives that are regulating sodium channels.
Thank you so much, Patrick. Can you hear me okay?
Yeah.
Okay. Should I just start sharing my screen? Let's see.